Hello. So today I will be presenting the current development of vaccines against virus infections, and then we'll follow that with a COVID-19 vaccine design. So the content, first we'll start with the literature review, and then we'll move on to the vaccine development. Um, so what are vaccines? Vaccines are a form of treatment where the body is introduced to either a killed, weakened, or a subunit of the germ so that the body will become familiar and produce antibodies and provoke the immune system to produce B lymphocytes, which are defensive white blood cells, to produce memory cells. And these memory cells remember the germ the next time it attacks. So when the body is exposed to the disease, the next time is quick to respond to and produce the necessary antibodies. There are many different types of vaccines. This includes the live-attenuated vaccines, the inactivated, which are the killed vaccines, toxoid vaccines, subunit vaccines, conjugate, mRNA, DNA, and recombinant vaccines. I'll be talking about all of them. So a brief history of vaccines. In the 17th century, a man called Edward Jenner, he exposed a patient with cowpox, and this cowpox it granted immunity against smallpox. So once this happened, his methods were like rose to fame, and eventually smallpox was eradicated, as you can see in figure 1 below. In 1980, smallpox was eliminated from the world. In 1885, a guy called Louis Pasteur he invented a rabies vaccine. This was way after the Edward Jenner. And this was also a great impact in vaccine development. And then in 1930, more vaccines were developed, such as vaccines for diphtheria, tetanus, and tuberculosis. So there are many different routes of administration for vaccines. The first is under the skin or the subcutaneous method, or in the muscle injection. As you can see in figure 2, the muscle is the last layer, and the subcutaneous tissue is right under the dermis. An example of this would be hepatitis A. Another route would be oral vaccination through the mouth. And an example of this would be tuberculosis vaccine. Also, there's a vaccination route through the skin, which is application of the vaccine in the dermis area as shown in figure 2. For smallpox, there's a method called saccharification, and it's not used anymore. So the last route of administration is through the nose, and an example would be the influenza vaccine. So what are the different types of vaccines? The first one is the live-attenuated vaccines, which is a weakened version of the real pathogen. And this germ is weakened by repeated culturing, which takes very long time, and it reduces the pathogenicity eventually of the pathogen and makes it so that you can put it in a vaccine and give it to the body. And since the pathogen is weakened, it has very small or minimal effects on the patient or the person that takes it. Also, the immune response, the best thing about this is that the immune response, even though it's weakened, is still basically identical to that of the real pathogen. Examples of this type of vaccine would be chickenpox, yellow fever, and rotavirus. Advantages of LAVs is that it has high immunogenicity, which means that that no need for adjuvants or booster shots. 1 or 2 shots can provide lifetime immunity for the virus. And also it produces essentially identical immune response, just as I mentioned before, to the real pathogen. Disadvantages would be that in rare cases, there's a reversal of the weakened pathogen to the original form. And also it cannot be given to patients with immunosuppressed conditions, such as patients who do chemotherapy or have HIV. Another disadvantage is the transport. You need to keep the vaccine refrigerated and stored to make sure the vaccine doesn't die. Another type of vaccine is inactivated vaccines. Inactivated vaccines are basically, like I mentioned, a killed version of the pathogen. Even though the pathogen is killed, the body can still pick up the antigens from the germ and store the memory cells for when the pathogen actually attacks the body. How do they kill the germ? They usually use heat, formaldehyde, or gamma rays to achieve the inactivated vaccine or the germ. Examples of this type of vaccine would be hepatitis A, influenza, polio, or rabies. Advantages of this type of vaccine, inactivated vaccine, is that it's much safer than LAVs since the germ is killed. It's also easily stored and shipped since the vaccine is dead. It can't give you any pathogenic effects, and it can be given to immunosuppressed patients, which is a huge advantage because it gives more broad people to take the vaccine. Disadvantages would be small immune response in the body, which means multiple shots are required to achieve optimal protection. Another type of vaccine is toxoid vaccines. They're basically, instead of exposing it to the actual pathogen, you just expose the body to the discharge of the pathogen, such as toxins. So an example would be diphtheria and tetanus. So what it does is that pathogen is the toxin from the bacteria is weakened or killed using heat or chemicals, just as like the inactivated vaccine. And then that toxoid is made into a vaccine and treats people. Advantages of this type of vaccine are very similar to the inactivated vaccine. The toxoid cannot revert to its original form, making it very safe. Lack of antigen multiplication, cannot spread to unimmune patients. Inactivated toxoid means easy transport and storage. It's not very sensitive to temperature, humidity, and light, which is very good. Disadvantages, small immune response, just like inactivated vaccines, since the toxoid is inactivated, and multiple doses are required to maintain immunity. Another type of vaccine is called the subunit vaccine, and this is where a part of the germ induces the highest immune response in the body. This subunit is chosen specifically, and it's determined to produce the highest number of antigens and to contain the highest number of antigens. And these subunits are usually proteins or polysaccharides. Examples of this kind of vaccine are acellular pertussis and hepatitis B. Advantages would be that it's very safe because the fragments in the vaccine cannot cause disease. They're not alive. They're only part of the germ, and they're suitable for immunosuppressed patients. Disadvantages would be the immune response was small, and that booster cell, because of this, booster doses and adjuvants may be required to maintain the immunity. And also the determination of the best antigen location and separating it from the actual germ is sometimes it's not possible, or it's very time consuming, and it's a major disadvantage. So another type of vaccine is the conjugate vaccine. What are they? They are basically used when pathogens. So some pathogens can trick the immune system by hiding their antigens with a coating of polysaccharides. So this is when we introduce a conjugate vaccine, and they basically produce a similar pathogen that the body recognizes and administer it to the body. The body links to this camouflage coating of polysaccharide. As a result, the immune response is higher. This conjugate is usually created using appropriately engineered E. coli in a cell culture. Example would be of this type of vaccine would be HIV vaccine. So advantages would be that it grants immune response to both infants and adults, and that it gives longer protection. And this is when compared to polysaccharide vaccines, which are just a subunit vaccine. And another thing is that there's no immunotolerance, which means that repeated shots give the same benefit as the first administered shot. Disadvantages would be that the preparation is complicated and requires much more steps. And vaccines can also have mild side effects, but that's very normal. such as swelling or redness. Another type of new vaccines are called mRNA vaccines, and this is where synthetic RNA is produced and placed in the immunity cell found in the body. This mRNA sends instructions to the ribosome to produce proteins. These proteins are programmed to be like the ones that the pathogen would produce, and that as a result would grant an immune response in the body and create antibodies. An example, a very common example, is COVID-19. Advantages of this type is that they have very quick production, cheap production costs. They have the real pathogen is not introduced to the body, which means it's very safe. Disadvantages is that it's new, so there's a lot of unknowns, and the FDA has more regulations since they are hesitant. Since only a subunit of the pathogen is produced, booster shots may be required to make sure the immunity stays at a good level. Another type of vaccine is the DNA vaccine, and these type of vaccines are a vaccine in which the gene for the desired antigens are found and cloned. The clone is then put into a vaccine that is given to the patient. This type of vaccine is still in experimental stage, so it's not available for human use. Figure 4 here summarizes the process of DNA vaccine production. Advantages of this type of vaccine: very stable, easy transport and storage, quick production, since DNA sequence can be easily changed in lab, large scale production at much lower cost than traditional vaccines. Disadvantages would be that it doesn't work very good with polysaccharide shells, viruses with polysaccharide shells. You're better off using conjugate vaccine for that. And another disadvantage is a complicated design and action. Recombinant vaccines are very similar to DNA vaccines, except here they're not grown in the cell, they're grown in a test tube. So they are live replicated vaccines that are engineered to have extra genes. And these genes produce the proteins that provide the immunity. The proteins are then used to make the vaccine. These vaccines are still in the experimental stage, like the DNA vaccines, and have not been approved for human use, although animals have. Advantages would be that it has easy production and scalable to desired production size, less risk of side effects when introduced to the body. Disadvantages would be high cost of production because antigens must be found, cloned, and then expressed. Also, like mentioned before, it's an experimental stage, so not as much development in this field. So now that we have the types of vaccines figured out, we can develop a vaccine for COVID-19. So COVID-19 is when a virus called SARS-CoV-2 attacks the body, and it produces the disease called COVID-19. The vaccine that we will be producing will give instructions to produce the spike proteins shown in figure 5 here. The red spikes here are the spike proteins. These spike proteins are harmless, but when they are introduced to the body will respond, and it will produce an immune response in our body, creating antibodies against this SARS-CoV-2. So SARS-CoV-2 stops the ability to make protective proteins while still replicating rapidly. NSP1 is basically what is responsible for the pathogenicity of the virus. Here in figure 6, we can see that the NSP1 here stops the mRNA from entering the ribosome and thus does not allow the proteins in the ribosome to be produced. So what is the goal? So now that we have the research, we can develop the vaccine. Our goal is to produce the spike protein to develop immunity, as mentioned. So RNA vaccines are the fastest vaccines to be produced, and with millions of people in need of vaccination, mRNA vaccines will be the chosen type of vaccine for our purposes, as the pros are much greater than the cons. The vaccine will be injected to adults over the age of 16 and in the upper arm muscle. Typical. Once injected, the mRNA is delivered to the immune cells to give instruction on production of spike protein. And then after production, the cell breaks down the instructions and removes the mRNA, eliminates all of them. So we determined a dosage of 0.3 milliliters is optimal, and 2 dose series separated by 21 days. Protection will save the patient from disease for up to 6 months after the second dose that is administered. So it's only temporary vaccination. Common side effects after delivering the vaccine would be redness and swelling of the injection site. Also, body side effects include headache, fever, chills, muscle pain, nausea, and tiredness. And this is a good sign that the vaccine is working. After testing on, so basically once we're done with the research and development, we can start with testing on animals. So we tested on rhesus monkeys, and we found that when the modified RNA was injected in the monkeys, the monkey expressed signs of spike proteins with a successful immune response from the body. The injected RNA showed complete prevention of lung infection in the monkeys, which is basically what the main problem with COVID is. And the test also indicated that the RNA injected caused production of antibodies in monkeys and mice. So antibodies are what we need against the actual COVID when it comes. And then because of this, we moved on to the next phase, which is human testing, because the preclinical studies were very good. So in this human testing phase, as we mentioned, we used 2 doses of vaccine, and we also administered them alongside placebo shots. And these placebo shots were, these shots, the vaccine and the placebo, were given to participants age 16 and them being healthy, making sure that they're healthy. Immunosuppressed patients were part of the trial, so that we didn't leave them out, as long as they had stable medical conditions. And after we did the testing, we determined that the vaccine was effective against 95% of the people, and the rate of severe fatigue was only 4% for people treated with the vaccine. And with promising results from the human trial, we move on to FDA approval. So to get FDA approval, we need to fill out a form called the New Drug Application, NDA, and submit it to the FDA. So the FDA looks at the safety, the efficacy, the pharmacology, the toxicology, the dosage requirements, and the production method. And all of this was approved by FDA. And so we could release the COVID-19 vaccine to the public. So what is the production method that we used for our COVID vaccine? So first of all, we need to create the DNA, and then from the DNA, we need to transcribe it into RNA and then make the vaccine. So how does the DNA made? It's made in large vats by the process of biosynthesis. Engineered E. coli cells give out trillions of DNA plasmids in just under 2 weeks. After we get the DNA plasmids, we do a purification process on these plasmids, and we filter out the solution, leaving only the microscopic loops of DNA. These microscopic loops of DNA are then linearized, straightened out basically, using restriction enzymes which cut the DNA to make it straight. So instead of making it circular, it becomes straight. And then the linearized DNA is then packed into specially made bags. And these bags are frozen to a temperature of -112 degrees Fahrenheit, very, very cold conditions. And then the DNA is tested to make sure all the strands are identical copies. Very strict safety measures. The FDA are strict on this. And the DNA is then transcribed into mRNA. So now that we have the mRNA, first of all, the mRNA is produced in large vessels with chemicals and enzymes, and it takes a couple of days. So, and this process usually takes 3 months, but it can be lowered. and each batch can make up to 10 million vaccine doses. So now that we have the mRNA is separated from the excess chemicals and the liquids. The mRNA is then packed in bags once again and frozen. And then finally, once we have the frozen mRNA, we can turn that into a vaccine for use for people. So what are the biosafety procedures for COVID-19? It has a biosafety level of 3, and the level is dependent on the microbes used. So basically, it includes all the safety measures found in labs with safety levels 1 and 2. Plus, it has the safety measures of level 3 as well. So what are some examples of procedures for level 3? You must have appropriate personal protective equipment. The biosafety cabinet must contain appropriate materials for microbes in question. Laboratorians are monitored and might receive immunizations for microbes they are studying, along many other procedures that are found on the website, the FDA website. So lastly, we'll talk about the price and market analysis for COVID-19. In 2021, the global market for mRNA vaccine was calculated at around $32 billion worldwide, which has increased because of COVID. And in the US, mRNA vaccines are estimated at around 15 billion, which is basically almost half of the world market. So, and we determined the price of the vaccine will be sold at around $18.50 per dose. Thank you. These are the references used.